international collaboration, but different areas of the world kind of host their own site, even though all the information is shared globally. So this is the one run by the United States Department of Energy, kind of like NCBI with the Gene Data Bank. But if you ever get lost on the internet and you end up like something like this, this is the European version of the Protein Data Bank. But you'll see the one that I think we showed you, one of the two structures that we published is also in the European one. Because so they share all the information, it's just how they present it. It's a little different. So I'm going to stay focused on this version because that's the one you're going to be using the most. All right, and again, we introduced this briefly to you on Friday. We're just going to kind of walk you through a few more things, just to highlight a few different features, and then you can get started on the scavenger hunt. All right, if you're brand new and you're just starting your research, most likely you're going to come up here and type in some term, and then begin your search. So Rob's a big fan of TRMDs. And you hear us talk a lot about them. If you do just type in TRMDs, they actually automatically bring up a couple different versions, or a couple different ways of searching. In the beginning, it's probably best to just do a basic search. It'll give you more options. But in the future, if you're doing a search and you come up with, say like 3,000 entries, you might want to start thinking about using advanced searching to help narrow that down. But thankfully, TRMDs are not, although cool and up and coming, they're relatively new in the world of PDB. So if I click on search with just entering TRMD, I only got 147 structures. I say only, but you could get thousands of structures. If you enter a word like insulin, you're probably going to get thousands of structures. That can be a little overwhelming. All right, so, and that has to do with basically, let me just go back for a second. Let's see what happens when I do hit insulin. So if I did a basic insulin search, yeah, I got 7,000 structures. That's a lot of structures. And the reason being is it's not just going to return structures for insulin. Anytime the word insulin comes up, it's going to return a structure. So you can have insulin-like growth factors, you can have receptors for insulin, you can have insulin itself, you can have insulin from thousands of different ways. Something that even influences the production of insulin might show up. So in that case, that's when you're going to start looking at more, is it in keyword, is it in the name, and you're going to start using more. And there's another where you can really kind of do like a Google advanced search. But for today, I'm going to keep it kind of simple, and I'm just going to walk through some basic options here. So when you type in a keyword and you get your list of structures back, over here, there's going to be ways to refine that search. So if you look, the first thing that comes up is the scientific name of the source organism. So they do mention two different organisms sometimes in these entries. So I'm just going to click on this one for a second. So you will see organism and then either sometimes expression system where they list another organism. This is the one where the gene actually came from. So this is the organism that actually produces the protein. So this is from Pseudomonas. What the scientists did is they took the gene out, and we talked about this, I believe, on Wednesday, how they make the proteins. So they took the gene for the TRMD from Pseudomonas, and they put it in that BL21 system, actually from BNL, that we mentioned. So this is a specialized system for making proteins. So it's actually this organism's protein. It just happened to be made in this organism. And this is probably one of the most common ones because this was made in the early 80s and everyone uses it. Sometimes you'll see other organisms there. The Rosetta system is kind of common. For eukaryotic genes, sometimes they use insects. But that's usually when you're searching for the organism, this is what you're thinking, like what species makes this protein. And for us, ours was in Anaplasma. That was our target organism. for TRMDs, but Rob and me are also working on other organisms. We've been working on myoabscessus. So there's actually, if I click this and then hit the search key there, now what I've done is I basically refined my search to only the structures from this organism. And myoAB happens to be probably one of the most well-studied TRMDs. Long story short on that one, it's related to TB. So it's related to the structure of the species that causes tuberculosis. It's in the same family. It's very similar protein, very similar sequence. But this one is a little bit easier to manipulate in the lab. It's not as harmful for humans. So a lot of scientists use this because if you're playing with or experimenting with TB bacteria, you need to be like in a BSL-3 lab. So this one's easier to work with. And this group's very interested in TB because it's a serious disease worldwide. It's the number right now. All right, so I'm just gonna hit clear all to reset that. Nope, I'm gonna go back, sorry. So I brought them all up. So these are all yours, and then if I just wanna look at the ones for anaplasma, there's only 2. That's why that little number in parentheses is 3KNU, 41GC. And now some of you might be scratching your head saying, I thought you guys solved the structure. Just be careful sometimes with that, and that it's how you enter the information into the database. So on our structure, we entered it as anaplasma. They actually entered it as anaplasma, but a specific strain. So ours is actually right here. And sometimes you'll see that there could be multiple strains for a species. So here's Mycobacterium tuberculosis. That's the bacteria causes TB, but this is from this strain. And here's a different strain, CDC 155. So they're the same species of protein. They just might be slightly different strains. So be careful when you're looking. You might think you have all for, but there might be several strains, especially with bacteria. So if I click on this one, our structure should come up, 6UEV, because that's the way we put it in. We didn't have a specific strain that we were working with, so we just kept it as a species. All right, so I guess I'll just go back to a basic structure and look at the information. All right, so here's my 147 structures. I'm going to click on the first one and just kind of briefly bring you through so that you know what types of information has been posted. Some of this is review from Friday, so classification. So basically, it's a broad category as to what the enzyme or the protein does. So this is a transferase. It's kind of a little bit of the Wild West as to what you put it in. Some people are a little bit more specific with their classification, others are a little less. So transferase is basically any enzyme that transfers something. So our TRMD happens to transfer a methyl group to another, from one protein to the TRN. From one chemical basically to the tRNA. So yeah, it's technically a transferase, but this can get smaller and smaller, this category. Again, I mentioned this is the organism that's from, this is the organism that is expressed in, and then they write here mutations. So is there a mutation in the amino acid? So sometimes you'll find proteins in the data bank. They have the wild type or the normal version, and then they have different mutations. So you could see how an amino acid change causes a change in the structure of the protein. Deposited, this is when the structure was entered into the PDB, and then released is when it was made public. There's usually a little bit of a gap here because some people hold their structures from public view until they get their literature published or the article they're working on. And that's down here. You'll see the article. A lot of times they publish a structure and the paper is still pending. So you might not have a link, but if you want to find the paper, this is the PubMed link. And this is usually over here. All right, the paper, a little abstract, and who the authors are. Just going to come back up here. This information, this is the validation. Over here, this kind of gives you an idea of how good the structure is. There's different measures of how well your model's fitting your data. And we're going to go through these eventually when we go back into Qt, but this kind of gives you an idea of how well they were able to fit their model to the data they had. This also gives you an idea of how good their data was. So they have a resolution of 2.45, which is not bad at all. You can get lower depending on the data you get and the structure you're working with. R values we'll talk more about later, but basically these are, again, measures of how well the model fit the data. And when we go through Qt again and we talk about refining the model, you're going to do several validations, and each time you try to see if that number goes down. Every time the R value goes down, it just means basically your model is matching the data better and better. All right. And then there's just a whole bunch of information that we're going to have you guys kind of explore and click on and try to answer some basic questions if you could find the information and learn a little bit more about these proteins. This does happen in the past, so some of these, if you look at someone's PowerPoint, it may have changed a little bit, but basically there's usually a summary page here where the basic information is given. You can click on this 3D tab if you really just want to explore the structure a little bit more. And there's some tools here. You can hide things and make things visible, change colors. You can scroll over the structure and they can actually tell you, I don't know if you can see this at home, but every time I click on, I'm not just
you can
mouse
see
over.
Yep, you can see it.
Okay.
Yeah, it changes
So
like a purple color. Yeah.
Yeah. So this is the one I'm highlighting, but if you look, as I highlighted here, it actually tells me which amino acid. I can do it the other way too. So if I want to figure out where 32, the 32nd amino acid is, it's a G. And amino acids have unfortunately, two different types of codes. There's a single letter code and there's the 3-letter code. So they use the single letter here, G, and I think it's glycine, but I'm surely a guess. And then it will highlight it in the structure. So I went too far. All right. So that's if you want to play a little bit around. I will tell you, if you're going to do protein structure biology, it is very handy to have a mouse with a scroll, which I don't have right now. I'm using my trackpad because my mouse died. And that scroll is kind of helpful in getting in and out. All right. So, all right. I'm going to skip annotations for now. Experiment. This is something you're definitely going to be looking at a lot in the future. So if you're doing, let's say you become part of a TRMD group. So this is 5WYR. Pseudomonas is the species. And under experiment, hopefully, if the authors did a good job of entering their details, they give you a lot of information about how they actually got the crystal to form. So they tell you the method. So vapor diffusion, hanging drop. So vapor diffusion is just a general method. And then hanging drop is, like we mentioned before, is the drop sitting on the cover slip, or sometimes you see sitting drop. Sometimes they put pH here. Sometimes it's actually over here. They left it out. So I'm not sure, but we could probably figure it out with a little bit more research. 293.15 is the temperature that it had it crystallize at. This is always by default in Kelvin. And then this is the actual formulation of the mother liquor. So this is the different percentages of the different chemicals that were in there. 9.9 times out of 10. This is probably some kit. So if you Google this, you'd actually probably find what kit it came from. Every once in a while, though, you also have people who actually get a slight hit. Maybe they get some sort of crystal to form, but not a great crystal. And that's when they start playing around with these numbers. Maybe 12.5% was too high, bring it down, or bring it up. Maybe, oh, there's the pH. So they put the pH by over here. 8.4, maybe that pH is a little high, try lowering it. So a lot of times they just get a lucky hit from the kit. But when we talk more about trying to get these crystals to form, sometimes you do have to play with these amounts to get crystals to form better and better. This is for physics people, so I'm going to skip a lot of this. But this will come up over here, space group. So space group is how the crystal forms, and it has to do with the way the repeating monomers kind of align or are in like I said, I'm not a physicist, how they kind of order themselves. So there's, I think, 100 and something different types of space groups. And sometimes, and we don't always know why, but sometimes the same protein, depending on the conditions you crystallize it, can form a different space group. That's actually one of the things that happened with us, with one of our TRMDs. Everyone was getting it in one type of space group, and for some still undiscovered reason, ours was forming a different space group. Probably because the conditions we used maybe were slightly different, or the protein was a little bigger, who knows. But that actually led to us being able to get some different information for the protein. It's almost like we were able to see the protein from a slightly different angle, and we got a spot on the protein that normally isn't seen. And then there's some more just specific information about the actual data collection, which we're not going to go through because that's a little high end right now. And there's the sequence tab. If you go here, this is recently within the last year changed. You could actually see the sequence of the structure. So if I zoom in, you can see up here, this is the amino acids. And I should be able to scroll aside. So this is just the first 100. Again, there are the letters, the sequence. This is it compared to the gene data bank. And then over here, they start to give you some information about the actual structure. This one, sometimes they'll tell you where certain binding sites are. So if there's a ligand involved, they'll tell you where it actually binds. They usually give you, this one doesn't. Again, it depends on the, oh, maybe it does. So this is the secondary structure. Sorry, I don't have it zoomed in, but you can see where the different structures are, like the alpha helixes or the beta sheets are. They actually did it differently. So they're SFG, which is the ligand actually. So they didn't put it up here. They have it down here. This is where SFG is binding. So it's kind of helpful as far as trying to figure out the active site. So this is where the active site would be. The amino acids down here. All right. So really, the best way I could tell, clicking around and trying to figure out. So what we did, actually, I'm just going to go to show you the classroom because I see that we're almost done. We put a little Google form in the database, and if you just put your email in there, and then we have little exercises. So I started with a basic search or TRMD. So when you do that, you'll try to figure out how you try to answer the questions based on those searches, and then click on specific structures and try to figure out, just so we know that you know where you're finding this information. So we have 3 different searches and a few different specific structures to see if you can find the information out of the PDB. But I didn't want to go past the time. But that's pretty good. Any questions?
I think they're good. So again, you're going to find that, as John said, on the classroom. You could work together on that. Each of you should be submitting an individual one, though, but you can definitely work together to try to find all the little bits and pieces in there that are on the in the PDB. So with that said, that's it. So you guys can work on that and have that done for Wednesday. On Wednesday, we'll take a look at what some people did. We'll review that a bit, and then we'll do a little bit more information hunting on the PDB on Wednesday. And that's it. So if you have questions at any time, you can email me and I can get some more information for everyone. And that's it. So again, everyone, thanks a lot. It's 10:50. So if there are no other questions, you're free to go and you can start working on that scavenger hunt and just have that ready to rock and roll for us on Wednesday.
Have a good one.
Thanks. You too, guys.